All right, welcome everybody to the February 3rd, 2026 edition of the New Jersey Mycological Association's Taxonomy Tuesday. The topic for tonight is going to be Leccinum and Leccinum related mushrooms. So we'll start with a little bit of the discussion about Leccinum species that may or may not occur here in New Jersey, and then we'll open it up for shared observations. So to start us off, I did my best attempt at trying to come up with a checklist of species for New Jersey. So I thought I would share that list to begin with. Um, see, can you see my screen?
Yes.
So
Yes.
we got three columns here: good names, provisional names, and then excluded names. And then under the good names column, I've got some asterisks on some species that their distribution is uh, not confidently in New Jersey, but we do have some records of people claiming to have things here. As you can see, there's basically three genera represented here: Hemileccinum, Leccinellum, and Leccinum. The boundaries between those three genera are probably fluid and, most relevant here to pay attention to the epithets. But as far as I can tell, these are the species that seem to be pretty good names for uh, applying to mushrooms in New Jersey. And then a lot of these excluded names over here are because they're European or they have records that we need to verify. So some of these we might be able to confirm tonight and look at some species or some records and maybe move some species from one column to another. But I thought this would be a good place to start. Um, any comments or input on this list before we get started?
What kind of input would you like to hear?
Well, do you think any of these are misplaced? I mean, obviously the provisional names are provisional names, but do you agree that none of the species in the excluded names are relevant to be applied to mushrooms that occur in New Jersey, and the things over in the good name column are things that could reasonably be found here?
Okay, let's start with excluded names. Well, I mean, you're basically saying Leccinum sensillado, Hemileccinum is not strictly in Leccinum, in the sense of cool, it's a separate genus, but yeah, they have sort of, these floccules or scabras, if you want to call them, but I would not include them in Leccinum, even in the broad sense. So Impallidium is out, that's European.
Right.
Crostipodium, probably also European, that's out, and so is Grisium. Arenicola is a North American name. That species is associated with beach grasses, sand dunes.
Mhm.
If it's proven it's a distinct taxon, but that's a legit name. And maybe we can find this thing on the dunes of New Jersey, coastal areas.
There is an INET record of this we could look at
Right. So
later.
again, they're not associated with trees. They are, sedges, or how you call them, grasses. These,
Yeah.
small, not even shrub-like, grass-like plants that flourish in sandy, not just in sandy soil, but in the dunes, in the coastal
Mhm.
areas, like next to the ocean, essentially. So you can transfer Arenicola into the good names section.
Neat.
Uh, Aurantiacum is a European name strictly, and that thing is associated with a broad number of broadleaf trees. So we don't have it in New Jersey. Um, Picenum might be synonymous with vulpinum, even though that species was found under spruce and vulpinum under um, pines. And the other species, I don't recognize what they are. They could be European, one of them, like leucopodium, and glutinopalens, I think, is probably North American, but I don't know what that thing is.
Cool. All right. So,
The good
um,
names.
any of these you can immediately throw out as being like, yeah, there's no way that's in New Jersey.
Okay, so aenigmaticum is my name as provisional, but it's going to be a published species one day, and I'm pretty sure it will be. So, but yes, that's in New Jersey. Hortonii is in New Jersey. Hemileccinum rubropunctum is in New Jersey. Hemileccinum subglabripes is quite rare, but it is in New Jersey. I have a sequence collection from Stokes, so that's still good. Albellum is very common. Atrus tipitatum, I think, is more boreal. I don't think
Mhm.
we have it in New Jersey, but it's a North American name. Holopus is good. We do find this in the Pine Barrens, by the way. Insigne is a name by Smith and Thiers, and I don't have a good understanding of that species. It probably grows with poplars. We might have it in at least PA. I think they found quite something that resembled Insigne.
Yeah,
That
Um,
grows with apple.
Nescutec in poplars,
yes, yeah, we,
something
we
that
we
looks
actually
like it
were
at
together.
least.
We were together maybe like 10 years ago, and we found that thing that looked like Insigne. But again, who knows what that thing is, because they're difficult. These orange red cap things are difficult. Leccinum longicurvipes is a good name. We have it in New Jersey. It's a common species. We do find it in the Pine Barrens as well. Leccinum luteum, finally we know what that is. I can show a collection of bona fide luteum. It's not what we thought it was,
Hmm.
but we got the type collection sequenced, and we do have it in New Jersey. We have a collection from Smithville. Pseudoinsigne goes with insigne. I don't know what this species is. It's might be associated with birch, but it's still a red capped or orange capped, I think, species. Leccinum rugosiceps, we probably have it, even though the concept is not readily understood. I think we do have it. Scabrum is European species. We don't have it in New Jersey. I haven't come across it. Um, snellii, that's a yes. Vulpinum, that's a yes. And subgranulosum, I don't know anything about that species.
Cool. All right. So that's the playing board we're dealing with tonight. Um,
And then, sorry to interrupt, you have all these provisional names. These represent um, operating taxonomic units that have unique ITS sequences. And whether we can match them to anything that Smith and Thiers have collected in Michigan, many decades ago, is another story. So this is a work in
mhm.
progress. But Indiana 01, Indiana 02, I think they're found in New Jersey. And of course, the New Jersey species 1 and 2 are there. VA01, I have to look that one up. And Leccinum, Indiana 01, I have to look that one up too.
So VA01, you have an MO record from Horseshoe Bend.
Oh, I know what that is. So that thing, what we thought was luteum, and it's not.
Okay.
Okay,
And
that's
then
the one
the
growing with hornbeam. Yes.
and then the Lexinellum, Indiana 01. You've got two records. One is Somerset County, other is Baldpate Mountain.
Okay, I'll look it up. All right. But again, as the name suggests, these are in the, they were so that um, um, cryptic code. Whenever somebody finds a unique species based on ITS, the code is given after the state where it was first found in. So Steve Russell happened to find it first in Indiana, and it was the first one of a kind, and that's why he gave it a code Indiana 01, and then it just goes sequentially after that. It could be other
Yeah,
states like New York 02, Virginia 01, and so forth.
Yeah, both Indiana O1 and New Jersey O1 were found at Freck.
Right, and my
Indiana
collection probably
O2 is
precedes the epithet. I didn't assign these things, so
hmm,
when I sequenced them, I just left them as Leccinum, section scabra, and Steve eventually came up with the idea of these um, codes, which is very convenient and very useful. And he just happened to match my collection maybe a few years later and gave it a name. So it just wound up to be Indiana one, even though in theory it should be New Jersey one.
yeah. And then Indiana O2 is a Stokes collection. And then New Jersey O2 is the most common, I think, of the provisional names, or at least the one that's most commonly assigned on MO. That seems to be all over northern New Jersey.
Yeah, it's a relative of snellii based in ITS2 region. I think I have a good understanding of that species.
Hmm.
It is very common.
So, I thought next we would just jump right into trying to review some of the, well, I don't know. Do you guys want to do some of the more common ones, or do you guys want to look at some of the more obscure ones that are potentially wrong, like glutenopalins and um, subgranulosum that we might be able to correct? And we'll start with the common ones.
Yeah, let's start with the common ones because some people here probably are not familiar with Leccinum at all. So it would be a good idea
Yeah.
to start with the ones that we can probably confidently identify.
So, Lila, do you want to start us off, or do you want you said you might be traveling, so I could walk through these if you're not able to talk. All right, so these are Lila's records. So her first one is uh albellum. Here are the photos. This looks like it's from northern New Jersey, maybe Morris County, Great Swamp area. As the name suggests, it's quite pale, white pores. This stem here looks less punctate than I typically think of Leccinums. I don't know, call it scurfy, or this looks more, peeling than punctate, but um,
Looks like it's mostly just damaged.
yeah,
Yeah, the skin just broke and stretched and cracked.
yeah,
But this one, this is Igor knows these way more than I do. He taught me this one. This one's pretty um, variable, right? Like it can have a
yeah.
pretty brown cap.
Yeah, it is variable. It can be almost pure white to sort of a color I can show later on in one of my observations. It can be very dark, sort of brown with some reddish tinges or purplish maybe even. And that's presumed to be the same species. Yeah, you can see here. It's often rugulose, so the cap is going to be wrinkled, but not like in a tight way. It's going to be sort of these very shallow pits, very broad too. You can see like the central photograph there.
Mhm.
No discoloration. So if something stains greenish on the cap, it's not going to be that species.
Oh,
It's not going to be, yeah. So that is one of them. It can be as dark as this.
Yeah, I've routinely find these dark gray ones, but like Igor said, no staining on any part
No
of
staining on any
Leccinum.
part, and these are strictly oak associated. So
Yeah,
you will
that's
not
an
find them on the.
interesting thing about these oak associates.
But this is not
a
a
lot
strict.
of
This is not a strict Leccinum. Again, this is one of these things that I would put outside of Leccinum in a strict sense. because none of the Leccinum from section Scabra or Leccinum section Leccinum are associated with oaks in North America.
Hmm.
There are birch, pines, poplars, yeah, and that's it pretty much.
Isn't there also a carpinus?
That's also in the
Specific?
Leccinum
Yeah.
group. It's not a strict Leccinum. So this thing is not a strict Leccinum, even though it's an Leccinum. I think it used to be called Leccinum albellum, but again, Leccinum is polyphyletic, so we have to define Leccinum in a more strict sense, and then take care of all the other subclades and give them other names for genera, for genuses. But yeah, look at
Okay.
under oaks. It's very common in pine barrens, and again, the morphology varies quite a bit. And maybe there's a chance we have more than one, but I think they're all the same based on what I know so far.
Okay. Next up, we got snellii from Lilo. Looks like a similar location, Morris County. This one definitely has more typical Leccinum punctate stem. You can see that they kind of get a darker color with maturity.
It's not punctate, it's scabrous.
Scabrous, right?
I recently heard them called whiskers, and I like that.
Yeah, I'm not sure this is snellii. Snellii
That's the
stains
wrong
red.
staining for snellii.
Yeah, snellii stains both blue and red. And New Jersey O2, which is a relative of snellii, also sometimes stains red, but more rarely.
There's
Typically
blue in the
stains,
base of this one, but this like
yeah,
gray staining coming up the stipe, that's what would this one be? This one's
I don't know,
interesting.
Dave. The cap was not consistent with snellii. I can tell you for sure.
Yeah, it didn't remind me of snellii, the cap,
Yeah.
that is.
Well, if Lyle saved it, I think this is from section Scabra, so there's a chance we can identify that through DNA.
Cool. This is from this year.
And she found this near.
Near Great Swamp.
Great Swamp. Okay.
Yeah, even though the staining is reminiscent of some of the species in section Leccinum, this one doesn't have the sterile flap, and it looks pretty young. Also, it's not really expanded. It doesn't have the sterile flap of along the cap margin.
Correct.
He has a note here that it could also be variicolor, but I'm not familiar with that name.
Yeah, variicolor. I only have one collection that was made in Connecticut, and it got sequenced, and it matched a sequence from Europe from a good paper. So we do have variicolor, but it looks more like snellii. But according to the authors, there's a ecological difference. One prefers more basic and drier soils, the other one more acidic and wet. Something like that. I have to look it up. I always forget.
So we would call this Leccinum NR dot snellii, near snellii, but not snellii.
Yeah, FAFF. Yeah, but I'm not even sure. Like I said, without the sequence, I'm reluctant to put any kind of name on that.
Yeah.
Yeah, I wouldn't go past just saying this is in section Scabra.
Yeah, that would be a safe assignment.
Mm-hmm.
And just on that previous AFF or NR note, would CF come into play? Like compare it to snellii? Does that also work?
Well, CF, yeah, I mean CF is used. If we said Leccinum CF snellii, then we're saying it's close to snellii and it could be snellii.
Correct. Yeah, the
But we're saying
AFF.
it's close to snellii, but we know it's not
But it's
snellii,
not
so it gets
snellii.
something different. Yeah.
Got
Right.
you.
CF is used when you're almost certain that's what you have. You have to compare with the type, and then you can probably give it a correct assignment, but you're almost dead certain it is. Whereas
Right.
AFF is related to affine, which is not the same but related.
And you would use that in a situation where you know it's not that thing,
Yeah,
But it's the closest you can get.
but it's
correct.
the closest you can come up. Okay, cool. Thank you.
Yeah.
Knowing where Lila collects, it is very swampy and acid along this trail leading from the Great Swamp that she's on a lot. But maybe she can answer to that.
Yeah, I mean, if Lila has a specimen, if she saved this a specimen, we can get it sequenced and try to put a name on it.
All right, cool. Let's see what else she's got.
So I don't think this is maybe one of the varieties of holopus, do you? I think those holopus have different staining patterns. At least, there used to be like 2 or 3 varieties.
Yeah, holopus can stain red and blue, but it's
Yeah,
it's
and
very
it stains
pale.
blue. Usually very pale. Yep.
My main concern is the ornamentation of the cap. It's not really holopus-like.
Okay.
Cool. All right, this one looks like it's same general area up near Great Swamp.
That could be just your albellum, the dark one.
The dark albellum.
Yeah, could be. Mm-hmm.
Look how linear those scabras are.
Yes. Well, they're ridges, and then I covered it with these tufts of hyphae that dense, and then that's what that scabra thing is.
Mm-hmm.
Yeah, there's
Right,
some
sure.
interesting staining under the cap, whether this is true or not.
There's no flap along the margin, is there? I can't quite tell.
It's a very narrow, under a millimeter band of sterile tissue.
Yeah.
But the cap doesn't look like it's in section scabra. I think this is just albellum. Whoa.
Reason I'm not logged in. Yeah. All right. And I think that's all that Lila had. So Hunter, you want to go next?
If there's, yeah, I could. I could. So this name was put on by INAT. I found it in mostly white pines, mixed woods. I was actually thinking this was a different one, but this is from northern Wisconsin, and it's was a little bit different than the red caps I had found before. The stem is really white, and then the scabers are rather scarce, and then there was some staining, kind of greenish. That name I'm not really familiar with, and so I just took it, but I just put this up like at 5 o'clock today, so I haven't had time to look into it at all. I was just wondering if anyone knows about this name.
Well, I looked it up. It's European, so that's
Okay, yeah. So
that's
it's
so far a
even
chart.
there,
Yeah.
right? So why is it I not giving me European names in Wisconsin?
Because INAT is full of it.
Yeah, INAT's not filtering their AI enough through location.
Well, it
apparently.
is, except that other people are assigning that name to North
Oh,
American
I'm
materials.
sorry.
So
Right.
if we were to go in and correct all of these occurrences, then it would be less likely
Right.
to apply that name. That's how it snowballs, because a few people will assign the name incorrectly, and then the AI starts to suggest it, and it starts to become a self-fulfilling prophecy.
Yeah,
Okay.
okay. I see, because it's user-generated AI. It's not technically generated
It's not,
AI.
yeah, it's not pulling
Yeah.
from, a central database of
Right.
high
Yeah, it's
quality.
user-generated. Well, anyway,
Yeah.
this thing reminds me of what we have sort of called insigne. Um, it looks like the thing that for lack of a better name we said, and is it insignia or insignia? It's insigne, right? Or
Right.
insignia.
Yeah.
Yeah, it's and it could be. I have another observation that I put that name on. Um I feel like the stipe is so white and also so substantial. But also we can't see what you keep bringing up, which is that margin flap.
Oh,
Uh,
sure you can. Look at the one where it's turned.
Yeah.
oh, okay.
Yeah,
Okay.
look at
That's
that
all.
the
No,
flap.
that's all sterile tissue.
Yeah, so
So
Yeah, that's the
that
flap.
is that would be insigne like, right?
Yeah, that one's in section Leccinum, I believe. This is just a
correct.
Yeah,
really young one. It's not expanded. The stipe is still pretty white.
yeah. It's hard to read. Yeah. Okay. So yeah,
Yeah, the bottom
I'm good
line.
with that. I'm surprised the name didn't come up, but yeah, I will correct it. I think I would agree.
Yeah,
Yeah,
what
you can mess
Jason
with the
is
name on
saying
this.
applies here. There's probably other collections in North America that look like this one. They're just very young and don't look like the older version. And like Jason said, it snowballs. This
Mhm.
name
Yeah.
gets applied once, then three more times based on that, and then there you go.
It's the same
Right.
idea with gem bank. Somebody posts a sequence, and then everybody just takes after that, and you have
Right.
a proliferation of the misidentified collections with a specific sequence. So to begin with, you have to start identifying the habitat. Was this found with conifers, or was found with birches or poplars? And I see conifer needles here.
It's mixed woods and oak, for sure.
It's an oak
Well, you
here
can exclude the oak because none of these things grow with oaks.
because of the sterile portion of the cap.
Yeah, I mean you can readily place this into sectional Leccinum. All
Right.
these
A
red and
lot of,
orange capped.
I mean, if you look up some of these Leccinums, it does say they grow with oaks. Some of these red cap.
That's incorrect.
Oh, okay.
I think that's mostly incorrect.
Okay, because it's definitely out there in books too.
Well, I
Um,
think doesn't Arenicola, the European thing, doesn't
Well,
that
that's the only one, but that one is
yeah,
very non-selective. It grows with essentially any
right.
hardwood tree in Europe.
And I have found red cap Leccinum growing in pretty much hardwood forests with conifers not very close. I think I might have one in my links. I'm not sure.
I think all of these are either birch, conifer, or poplar, aspen.
Yeah, I
If
would
it
I would
has
guess
this
poplar.
sterile, if
okay,
it has
so
that sterile tissue around the cap, right, Igor? If it doesn't have that sterile tissue around the cap, then it could be an oak associate. Is that
No,
what you're
it's a
saying?
pronounced, all sectional Leccinum have a pronounced sterile flap, sterile tissue flap on the edge of the cap, unlike all the other
Right.
lichenoid fungi. And as this thing grows bigger, these flaps are very prominent and very irregular. You'll see them later on in some pictures.
So but there are lexinoid mushrooms that associate with oak, except, because of this sterile flap, we know that this is in the section that doesn't associate with oak.
Yes. Well, not only
Okay.
because of the flap, also because of the cap color and overall
Right.
morphology. I mean, this is like a typical sectional Leccinum presentation.
Right.
scabers that are white to begin with. I mean, if I found this thing in Europe, I would have called this Albus tipitatum, which is a legit name, like this one here. Albus tipitatum from Europe. The scabers are white. They're not even black or brown. But
Even this young?
yeah, I mean, typically, I mean, when they're young, chances are that it's going to be all white or mostly, but not always. I think it varies. I think I have no idea like what really is responsible for the color of the scabers, you know, in older age or younger age, and all the variability. But it can be white, it can be brown, or it can be black, and a mixture thereof. So I wouldn't really dwell on that morphological trait because it does not going to get you anywhere.
Yeah, look at the gradation of the color of the scabers as you get
Yeah, you
closer
see,
to
it's
the top.
brownish, it's grayish, and it's white. So
Yeah.
you have all of them, you know, in one fruiting body.
Yeah,
I see
cool.
conifers. I would probably say this is in the vulpinum group, unless you saw birch also and poplar, then it becomes more difficult.
Yeah, this looks like the thing I see sometimes in poplar, you know, not all that commonly.
Okay.
Yeah.
Well, and I just threw these up. So I mean, if someone else has some better observations, you can see I didn't even put a location here. I know it's from Wisconsin, but if there's some better observations, I'm happy to like wait till the end too. Just saying.
No, let's see what else you got.
Okay. So this one does I think is insigne. It does have that, you can see the flapping a little bit. Right?
Well, they all have flaps. All sectional Leccinum have flaps.
Okay. Yeah, go ahead.
Was this found near where the first one was found? Because I think it's probably the same species.
Yeah, I agree. And no, it was another trip, I'm pretty sure. Another spot.
Okay. So maybe it's different species. I thought maybe they were.
I'm telling you,
No,
they all kind of look the same. You cannot really
Yeah.
make a distinction. I mean, it's just it's a
Yeah.
disaster.
I mean, that's why, you know, I started to put a presentation together that went through all the species, and I'm like, you know, it's 20 slides of the same mushroom.
Yeah, like in the movie, there's a forget about it and there's a forget about it.
Funny.
Yep.
But
Some sources. Oh, God.
I thought the color of the stain, I mean, can help sort out these.
Not really. Not really. Don't even dwell on that. It's you know, they all stain blue, or sometimes they don't, or sometimes they stain pink, and then it becomes more violet gray. You know, there's going to be all sorts of colors, and they're not consistent with any species. So you can just throw it all out the window. It's useless.
Okay. And so, and even tree species, it's a little dicey because, I mean, you can't, that isn't up for sure either necessarily. I mean,
Well, if you're finding your Leccinum section, Leccinum in a grove of poplars, then you can eliminate a bunch of other taxa that are birch associated and conifer associated, right? Like vulpinum, you can throw out. Because again, remember that Leccinum are mycorrhizal specialists, okay? Some taxa are going to be only growing with a particular tree genus or species, whatever. Like, you know, all these scabras, section scabra are only birch associated. You will not find them with any other tree. All right.
so that's
These guys can grow with pine, poplar, and birch.
okay.
But again, there's going to be a species separation between those hosts.
Mm-hmm.
But you have to figure out which is which.
Right. Okay. But it helps to know the trees.
Right. For instance, if you found this in a strictly deciduous woods, probably with birch or poplar, and there were no conifers around, you can eliminate vulpinum for sure.
Wouldn't it be good to add to the New Jersey list if some of them are, distinct with certain trees, to also name the tree? on the list that Jason had at first.
Yeah, yeah, we could do that. I did some of that already. Let me see. So
At least in future. I know there's not any room right now.
yeah, I mean, not all. I mean, I just went based on the Bassett's, um, the Bassett's book, which didn't have all of the species. Um, but I'm curious which one. Like albellum, is an oak.
That's an oak, correct.
Um,
Subglabripes can grow with mostly birch, especially in the northern range of it. But you can also, I think it's mostly birch associated.
I've found subglabripes in a variety of different habitats, but although that's probably more than one species, so maybe I'm just finding different versions of subglabripes.
Yeah, we have at least two more candidates for subglabripes, as it turned out, through Steve and Colin. But I think, in my understanding, subglabripes is a boreal species that likes birch. And I'm pretty
Okay.
sure, yeah.
How about hortonii? I would say oak.
That's oak.
Hickory. Oak hickory, I would say, for hortonii.
Rugosiceps I have as oaks, especially pin oak.
Yeah, but that's your Leccinum, so that's not strict Leccinum. Yes, we can put down oak for that one.
Holopus is a birch/hemlock.
No, holopus is birch only.
Birch only. Yeah,
Yeah, sometimes there's hemlock where you find it
nearby.
but
Yeah, with
but
birch,
there's always birch
associated
there
with the birch.
Yeah, this is section scabra, so remember, all section scabra is birch only.
And you have to be careful with this Leccinum luteum thing, as Igor pointed out. It is not what we thought it was, and I have one of my links goes to something that was sequenced that we thought was luteum, and it's something else. I forget what it is.
I think luteum and VA01, both of them I think are hornbeam associated.
Right, that's the Carpinus, the one that's listed as Carpinus carolinea, the musclewood or ironwood.
Oh, that's cool.
Snellii is also birch exclusively. This is section scabra.
And Carpinus is in the same family as birch.
Right.
So, yeah. Let's see what else. Vulpinum is the only one that's a conifer associate. Is that true?
Pines, yes.
That's the only one.
Right.
Yeah, and that section Leccinum, that's your typical, orange, red capped, robust Leccinum with whitish or gray, dark gray or brownish scabers. Yeah, it's very
Vulpinum is
some of
mostly
these
pine, but sometimes Norway spruce.
Yeah, there's a problem here with vulpinum. I think some of these provisional names, maybe like IN01, I forget. I've got one or two of them in my links. They look like something that would be called vulpinum, in sort of a field guide sense.
It's IN02, I think. Yeah, this is Steve's cryptic names, and I think he applied that moniker on a bunch of different tags. Like I said, ITS and Leccinum section Leccinum is just a disaster. You can just forget about it.
Yeah. So yeah,
longicurvipes. Is that an oak or is it oak pine?
Well, it's found in the pine barrens. I think it is a. I think it's a hardwood associated species, probably oak.
I have found longicurvipes where there are no conifers, but I usually find it where there's a mix of pine and oak.
Yeah, we find this in the pine barrens. I think it's oak associated.
Yeah, I would not disagree with that.
Yeah, and I think Colin Damnauer specifically wants to put that species into a different genus.
Wasn't that Hemileccinum for a while? But I guess it's genetically doesn't fit into
It
Hemileccinum.
doesn't fit. No. I mean, it's completely outside of Hemileccinum.
All right.
It's closer to Leccinum than Hemileccinum.
This is another hunter record from Wisconsin.
Yeah, and another one I just threw up before, which is why I was saying if anyone else wants to go, because I don't really have much to say. It looks like there's some brown staining on the pores. Um, uh, yeah,
Yeah,
and
they do that. Yeah, that's very typical of Leccinum.
right. So, and I mean, to me, it's not really red capped, but it probably is just another.
It's an orange brown. Yeah, it's kind of muted tone, but you can see the irregular flaps. So you know this is from section Leccinum. This is not section Scabra. And again, habitat information is very important. Was it aspen? Was it birch? Was it conifers, etc.? You have to start from there.
Yeah. Okay.
Yeah.
So, so how many sections are in Leccinum?
Well, it's difficult. Like I said, they lump things together, but Smith had his own taxonomic scheme for Leccinum, which is now completely broken down because Phylogeny doesn't support it, so you can throw that away. It's still work in progress, but you can definitely have two well crystallized sections in Leccinum sensu stricto, in a strict sense. That section Leccinum, like this thing, and the birch associated gray/brown capped, more less robust, more elegant looking species that are only going to be birch associated. So those are clear cut sections. Everything else is up in the air.
You call that scabra?
Scabra.
Scabra. Yeah. Okay.
Yeah,
Okay.
and there's also a taxonomic scheme that further sort of breaks it down such that section scabra becomes subsection scabri. That's with an I at the end. But again, this is all up in the air, and I wouldn't even go with these more exquisite subdivisions.
Yeah, and I would say about the cap color of this one, it's pretty well expanded. So the color, the pigment's a little more spread out. So this may have been more vividly orange when it was
Yes,
like that one that we looked at first.
right.
Yeah. Okay.
So yeah, they kind of become, kind of brownish red, sort of the dingy or brownish orange. It's kind of a color that looks more brown than orange or red, but it's a light color. It's not as dark as the ones from section Scabra. It's a different tone of brown. I mean, if you photographed the disc, we would have gotten probably a better idea of what this thing is in terms of the color.
Cool. All right. Thanks, Hunter.
Okay. Yeah. Let's move on. Yeah.
And again, note the color of the scabers. They are brown here.
Right.
Okay.
Dave, you want to go next?
Oh, I have more. Actually, if you want, but we don't have to. It's like loose in the email. We don't have to do it, but it's a nice.
We'll come back to that at the end.
Okay, sounds good.
Yeah, this is a pretty cool one. We rent this cabin in the Adirondacks sometimes, and we'll stay for a few days. So this is over the course of 2 or 3 days. I forget the dates are listed in the observation, but this is one of these things that may have been called vulpinum or Piaceum, but it was sequenced by Steve Russell, and he put the provisional name IN02 on it. But as you go through the photos, if you just dash through them, you'll see them growing actually a little bit every day, and you can see also how the color of the cap does change a little bit as it gets older. No, that was when right as soon as I cut it, there was no staining immediately, but after a few minutes, I think the last photo shows then there's some staining setting in. It had this like fuchsia sort of stain setting in. And there, then there's the last one. It got even darker, right? And a little bit of blue in the base, also a little grayish blue, sort of.
It was 40 minutes.
So,
that's 4 minutes.
That's
So
very typical of
immediate.
Yeah.
vulpinum. That's what vulpinum does. It turns pink, then sort of fuscus, which is violaceous gray, and there's going to be bluing on the bottom of this type very often.
Yeah. By the way, I think the only trees in this area were fir and cedar. But where we rent the cabin in the Adirondacks, that's all fir and cedar, I think.
So the needles on the ground.
Oh, there were a few deciduous trees. There were like some maples or birch or something.
Yeah, that first photo showed, I think, some good needle cover.
Yeah.
Yeah, I think that those might be cedar, or maybe fir. I don't know. They're trees that I don't see very often because we don't have
this
them
could be
around
fir.
here. Okay, fir, right.
Also, the mosses there, that's pretty interesting. I mean, it's not uncommon to find these things with, in mossy areas, but
This was right near a lake, also. Not far from a lake.
Yeah, this is a boreal setting, and I think these are vulpinum-like species.
Yeah, that's why I brought it up about vulpinum being a name that it's hard to tell how to use that name.
Yeah, I would say it's a vulpinum or vulpinum group, and I think Steve's Indiana O2 species, it covers not only vulpinum but other taxa in sectional Leccinum, so that name itself is polyphyletic or whatever, paraphyletic.
Oh, interesting.
Yeah, again, he tried to align these sequences, and I don't think he just interpreted them correctly. So that cryptic code covers a bunch of taxa. It's not a single species he covers.
Okay. Well, yeah, I wouldn't have guessed that. Thanks.
And Igor, I've been wanting to ask when we're talking about these tree associations and ecologies, if you find one of these things in the pine barrens, is it most likely vulpinum?
Yes, I would say that. Yeah.
Okay. Yeah.
Vulpinum/vulpinum group in case we have more than one, and that's yet to be determined. I mean, you have to deal with the type collection to begin with because vulpinum was described from Europe. But I got, I sequenced many of them, several, and some of those ITS haplotypes were completely matching something from Europe. So it's probably circumboreal species, just goes all over the temperate zone across the globe, Eurasia and North America. And again, the evolutionary history of these things is very complicated. There's been a lot of introgression and hybridization, and then they split up. So you'll see a lot of different ITS sequences that all probably represent the same species, and then they speciate themselves, so it just becomes a one giant mess.
Cool.
So, but here, conifers, I would go with just vulpinum group. Definitely not insigne or versipelli or atrostipitatum. So you can exclude all these taxa. I would go with vulpinum. Vulpinum or Piscianum, if that name is not synonymous.
Did I propose a name to begin with beyond section before I got?
No, you didn't.
I might have to be logged in to see that. Oh, there it is. Yeah.
So
I just said section Leccinum. Oh, there you go. Vulpinum. Yeah, for Igor.
Versipelli's not on the New Jersey list. That's a northern one too, or something. Or what?
Versipelli is a synonym for atrostipitatum. Is that right?
That is very, very questionable. I would not merge these names right now.
I think Index Pungorum might merge them, but yeah.
Yeah, but Index fangorum still has Boletus separans as anthaconium.
Right. Yeah, let's see what the next one is.
Oh man, did I just hear that Index fungorum isn't actually something that I can rely on? That's
Occasionally,
troubling.
with a grain of salt.
They're very, very slow to update
Yeah.
update their names and their synonyms and so forth.
Yeah. So Dave, you and I were together when we found this beauty.
Oh, is this one?
Pinchot
Oh, okay.
State Forest.
Yes, that's right.
I remember the
Yeah.
spot exactly.
What did I have in the notes there? Because I don't remember the exact location. But it's the section Leccinum thing.
It's Pinchot State Forest. It's the Manny Gordon Pavilion area.
Yeah. So this might be growing with spruce actually. There's a lot of spruce.
No, this was all
there.
deciduous.
Okay. Oh, this is one that I meant. Yeah, this was one of these like strange ones. It's section Leccinum. It's got a red cap, but it grows in hardwoods.
Yeah, it's you say in the notes it's birch and poplar. So we saw both trees in there. We also
Okay.
saw oak, which is irrelevant. But this is your prototypical Leccinum section Leccinum, and look at the staining. And Steve Russell gave it the cryptic name of Ohio 01. So I think it is a different species. I'll take that. Which one though? Good question.
Well, he said in the comments for the last one, Leccinum IN01 and Leccinum Ohio 01 may all be the same.
But he was looking at all these sequences and he went for the same procedures. And I think the species is going to be different. Look at the staining itself. There's no fuscus. It's all sort of blackish.
Yeah,
And
the
there were no
habitat is completely different for
Yeah.
the two that
right.
we
There were no
see.
conifers. It was birch and poplar. So vulpinum is out.
Mhm. Cool.
Yeah, I mean, we still have the specimen. I think maybe Dave has half. I have another half. I have to look at that because I think I have my own observation. Or
Well,
maybe Dave took this.
I sent something to Steve. I may have sent him half the mushroom, so I might not have this anymore. But he sequenced it. That's why he's got
Right.
this.
He got the
name on it.
barcode.
Yeah.
Colin Damnauer told me that he's not going to work on Leccinum. He did admit that it's a very controversial and difficult genus. And he also said that somebody else works on these things specifically, and he's not going to interfere. He would rather focus on other things, in Boletaceae, as opposed to getting, stuck in this quagmire of Leccinum. And he's not dealing with these things at this time.
So who's working on Leccinum?
I suspect it's Beatrice Ortiz Santana. And
it's right, Santana and Quo, right?
He did say it was a she, so he didn't say it was a man. It was a woman.
Okay. Oh, you know what? I think Michael gave up on those. Actually, he did what he was going to do and left it at that. And yeah, it was Santana who was she a few years ago when I was putting stuff in for Fundus, she expressed some interest in these sorts of things. Right.
I mean, that's my understanding, my bet, but I could be wrong. I mean, Colin again did not mention any names, and I didn't ask.
All right.
Yeah, so you're right, Igor. This is the thing we thought was luteum, and it's the VA01. Yeah, it turns out to be not luteum.
No, not luteum, not by DNA.
That's a pretty one.
Yeah, so this one has an all yellow cap. It stains black when you cut it, when you stain it. So it's an interesting species, but of us, we got the name wrong. Which is very common for everything that Smith and Thiers described from Michigan, because that book was groundbreaking, and at the same time that book was a dead end, because nobody could figure out what these things were. I mean, everything. I'm not just talking about specifically of Leccinum. People just got stumped with descriptions and names and classifications, and that book basically, it's a classic book. It's a wonderful book, but it's of no use, little use.
I have that book. I like their books. I mean, they had great photographs for that time. I mean, really.
Yeah, they're black and white, and they do give you an idea, but a color would have been much better. So go ahead, Dave.
Oh no, I wasn't really going to say anything else. I mean, the habitat lists here a bunch of different trees. I think I know the area where this was found, and that's a pretty good characterization of the trees that are there.
Based on what I know, we used to find this under hornbeam, according to Luke, because we have a collection from Horseshoe Bend, and Luke was the one who found it, and he said it was growing under hornbeam. So,
well,
but
I might be calling the hornbeam beech in this setting. Because
okay,
I'm not sure I would know just from looking at the tree in the woods.
Yeah, 100% certain this is hardwood associated, because this fits the Leccinum in a broad sense. This is not Leccinum. It's going to be in the Leccinum group. So this is not your section scabra or section Leccinum. These things are very different.
I was
They
going to
are
ask,
more
do they stain?
longicurvipes and albellum like than they are scabrum or vulpinum like.
And do they stain, Igor?
Black.
Mm.
Brown, black.
You see a little bit of staining around the margin of the cap here.
And when you cut them on the flesh as well, on the surface, they all stain. No, I don't think they stain red, but only blackish brown, grayish brown. Yeah, you can see it there.
See it there on that injury on the lower stipe in the one photo.
Mm-hmm.
Those are just beautiful. I would love to find these. What's Smith's first name?
Alexander.
Yeah, so I've got a couple books. They're in color, the photos.
Maybe the later ones were in color, like in the 80s. I think he died in 1986, but the Boletus of Michigan, I think, dates back from 1971.
Okay. Yeah, I've got
the
Mushroom Hunter's Field Guide, which is like for Michigan. And then I've got the A Field Guide to Southern Mushrooms by Alexander Smith, and they're both in color. They're pretty nice. I just wanted to make sure I had the right Smith, because I was like, wait, mine are in color, so thanks.
Yeah, that's the same Smith that did work with Hessler, right?
Right. And his wife was also a mycologist.
Yep. Yep.
Smith Weber. I think she had a double barrel name.
Yeah, Nancy.
Yes.
I'm trying to find mine. I know I have. I think I have that same book hunter, the from Michigan.
Oh, cool. Yeah, I like it. Yeah, and my dad had it. I mean, this is like the oldest book. Yeah, I love it actually. So
this thing I do not find where I live in Pennsylvania. It probably does grow in some parts of Pennsylvania, but I've never found it around where I live. It really does smell like an ashtray with crushed cigarette butts in it.
Yeah, that's Hemileccinum, which is not Leccinum. It's a.
Yeah.
They are actually, it's in a different subfamily. So they are these things. Hemileccinum sits in the subfamily Xerocomoideae, with Boletus and Xerocomus, and so this is far away from Leccinum or Leccinoideae.
Yellow pores are a reliable way to most of the ones that we've been looking at have had white pores, right?
This looks a
Yeah.
lot like subglabripes too.
No,
Yeah.
this one is rubropunctum.
Similar.
So
Yeah, no, I know, but they're kind of similar.
they are similar. Yeah.
And I think longicurvipes can also has pores that get kind of greenish or yellowish, but that's also not strictly a Leccinum, or perhaps it shouldn't be.
Mhm.
Yeah, I mean, and these like really barely have scabers. I mean, it's like it's almost more punctate. I mean, they're just not really scabers, right?
Yeah, this one's just kind of roughened, really. This or even like Pharanos almost. It's just sort of got a grainy sort of a finely grainy sort of ornamentation on this type.
They are scruffy. There's like all these
Scruffy is the
miniature
word.
scabers. They are like scruffiness. Yeah, they're points of.
Yeah.
And subglabripes, it's even smoother, I think.
Oh, it varies a lot. I found subglabripes with punctate stipe, with smooth stipe, but like we said a little while ago, it's probably more than one species going under that name.
Mm.
And Dave, you mentioned the odor. Um, have, without the odor, would you have been able to ID this? Because sometimes I find these things and they don't have that. Um, so I'm wondering if
Well,
that's as reliable as reported.
I don't I don't find this species very often. I find it at Coma. It's pretty much the only place I find this. If I was to find one of these someplace else and there wasn't somebody telling me that it was what is it, rubropunct, is that the name, right? Rubropunctes?
Yeah.
I would probably call it subglabripes if it didn't have that odor.
Same.
Hmm.
Yeah, this Lila and I found this in New Jersey. We found more than one specimen of this, but I haven't I don't know it here at all. But that smell, yeah, it's pretty
Yeah,
interesting.
because the smell, I mean, it hits you. It's unmistakable, but I'm pretty sure sometimes with a lot of mushrooms that have reported odors, sometimes it's just not there, but that doesn't mean it's not that mushroom.
Good point.
I don't know. That's all I have to add on that. Like Eeyore, if Eeyore is still here, does that
Oh, I'm
mushroom
here.
sometimes does it sometimes present without the odor?
This one, yes. It's a mixed bag. I think some collections the odor is indistinct. I mean, my nose is not really that keen to begin with, but I can smell the ashtray when it's present.
Are
But
things in
you would find
general?
this only under oaks, whereas some varieties, the ones that I found in New Jersey were growing with birches.
Yeah, you know what? That's a good point. I don't think I've ever found subglabripes in oak dominated woods. Birch,
No,
hemlock, yeah, mainly birch and hemlock.
same.
Are things in general less odorous during a drought? I thought I remembered somebody told me that, some things just kind of, that they, that when they have a characteristic smell, it may not be as prominent if it's trying to eke out a fruiting body in a drought. But I forget where that's coming from.
Yeah, I haven't heard that. That's an interesting idea.
I haven't heard that either, but I will add, dehydrated collections of odorous material kind of stay odorous in my
Right.
experience.
Well, I
Not
mean,
sure if that,
Yeah,
I mean,
Go ahead.
My guess was that if there's a drought, then it may be putting the available water towards the fruiting body, and it might not
Yeah,
be producing the
just
volatile compounds or whatever
yeah,
is
that
making the
makes
odor to
it makes
begin
sense,
with. But
right? It's just
it's
putting
one of
all
those
the
things where
energy.
it's like, yeah, it makes sense, but I've never heard anybody actually like verify it. So it's probably just one of those things that gets repeated because it makes sense, and nobody's actually proved it.
Yeah.
Well,
Yes. Oh, go ahead.
one time I found chanterelles in, it was either Oregon or Washington State. I forget which one. And it was in August. It was dry. There was hardly any rain, and they were quite fragrant. So,
Mhm.
I mean, that's one example that I can bring up.
Mhm.
And
All right.
and
See.
I'll counter that just anecdotally as I never get apricot smell out of chanterelles. So even in good conditions. So
And I think it was obvious, and Brandon started to allude to it that I mean, dried mushrooms are more potent smelling than fresh.
Right.
But I know that Jason's theory, is separate from that. But just adding to make sure if people didn't know.
All right.
Nice picture.
Nice xanthoparmelia here in the background.
Yeah, so there's one area where I find these, not every year. Actually, there's two areas where I find these, but one more so than another. And some years there's a lot of these. I've seen, patches of like 20 or 30 of these. And you can tell when you have it has that really deeply wrinkled cap.
Did this used to be in a different genus?
Yeah, it's been kicked around. I think it was it Siracamus for a while? Siracamus?
It was like
I think it was for a short while. It was Boletus, if you go back far enough.
Yeah, that's how far back I'm thinking. So,
Yeah, and
okay.
it's not, yeah, but I think that went to
the
Siracamus for a while.
Interesting. And now it's Hemileccinum. Wow.
what is this, a KOH reaction on the cap surface? Oh,
Yeah,
ammonia and KOH.
ammonia and it's almost the same.
So, are these edible, Dave? Do you eat these?
Yeah, they're all right.
What about the ashtray one? Do people not eat that? Because
I would not try to eat that one.
yeah, I was just curious. Because the subglabripes are really good.
All right, and then these last things. Oh, there they go. I guess it did work.
So I just thought this was the coolest picture here. I
So,
think this is holopus. I'm not sure, but it's one of these bluish, greenish, reddish staining section scabra things. This grows with gray birch, but I just like the way this leaf stuck to the cap and almost looks like a fossil or something. And I think the next picture is once the leaf was peeled away. So it's just
Yeah,
I just like these photos actually.
and
Yeah,
and
then
it's a birch
the leaf
leaf.
is peeled away. Yeah, it's a birch leaf. Yeah. Um, so I used to find a lot of these section scabra things in this location, but they cut down most of the gray birch trees along this fairly wide trail because the trees tend to sag in toward the trail, and it's a state game lands, and they need to have access with a vehicle. So they took down a lot of the birch trees because they were falling on the trail and leaning over and getting in the way. There's still some birch trees there though, and there's some young birch trees, and I still find some of these section scabra things there. So I'm not sure holopus is the best name for this.
No opinion on the ID, but that photo is so cool with the birch
Yeah.
leaf on it.
Yeah.
Cool.
So this, I think, is from my yard, and I'm not going to find these in this spot anymore because we had to cut down the gray birch trees that were hosting this particular fungus. We had to cut the trees down because they were threatening our house. They're getting pretty tall and leaning toward our house, so we had them cut down last summer. And I didn't regularly see these mushrooms growing there, just since we've been there like 25 years, and I think it's only twice I've seen fruitings of these in that spot. But this one year there were a lot of them. So you can see the reddish, pinkish stain in the one on the right in the cap, and then there's like a bluish stain in the base of the stipe. That's a typical snellii thing. Now, is this actually snellii? Well, maybe. I don't think this was sequenced.
Cool.
Great observation, though. Really nice.
Yeah. All right, so either fibilosum or subalpinum.
Yeah, this is
From Montana.
fibrillosum. This is fibrillosum. It's hard to say. This thing is really, really dense. It's a Rocky Mountain species. And I suspect that some of the reports of people getting sick on red-capped Leccinum, and the picture is a little bit dull. It's not, the color is not coming through really well. It's a little more red than this. I mean, not a real vivid red, but these are probably, I mean, they're edible. I think I ate this one actually. But very, very dense flesh. And I imagine if it's not really well cooked, it's probably kind of hard to digest. So it's a Rocky Mountain species. This was found out in Montana, north central Montana.
Cool.
Cool.
Yeah, I'm pretty sure this is fibrillosum.
Mm-hmm. Right. Does anybody have anything they want to share from their screen?
I mean, I have lots of Leccinum observations, and many were sequenced. We can take a look at a few. I mean, it's unless somebody else wants to go, ahead of me, somebody who wants to have their collections identified, then I'll be at the very end.
I mean, I got some INET, not mine, but just some things that we can kind of look at and try to correct.
Yeah, let's do that instead, and we can always look at mine later on if time permits.
Can I put one in the chat? I think it's a
Sure.
Leccinum
Yeah, I'll share it.
thing. Igor, I think you've probably seen it, but it's
Skull
pretty
and
cool.
Run.
Yeah, Skull and Run.
Yeah, I think we got it sequenced.
I don't. You got Maricel's on the sequence, right?
Yeah.
Cool. Well, I'll pop mine in the chat if we want to look at it real quick, and we can maybe talk about it.
Sure.
Yeah.
I can share it.
Yeah, I'd like to see it.
Me too. Ooh.
Yeah, so this is one of those, Skull and Run isn't the Pine Barrens proper. Um, in our outer coastal plains, there's pitch pine. This was growing next to a pitch pine, probably, but a bunch of different trees, all densely packed. Look at how pitted that cap is.
Mhm.
So I think we were talking about rugosiceps at the time for this one.
Probably is.
Yeah.
But this still has to be confirmed. We have to see how many species that we have that sort of look that way. And in this case, I think ITS is going to be very useful, or any locus is going to be useful for barcoding. This is a good genus, or maybe it's going to be more than one genus. So yeah, I would say I would call this rugosiceps because it is North American. So yeah, it was very surprising because the habitat there that I saw was very pine barrensy, and we've seen not only this species but say Lanmaua pallidorosea was also there. So I was surprised to see it there because if you go to say places like Wharton State Forest, the heart of the Pine Barrens or the Pine Lands, you would never see anything like that. This or Lanmaua, they're completely absent, I would say, from the greater Pine Barrens. But there are probably going to be pockets within the Pine Barrens, niche pockets or habitats that might have a sprinkling of these upland, uh, you know, hardwood species growing inside the Pine Barrens. But as you said, Skull Run is on the outskirts. It's an intercoastal plain. It has a sort of a character of both the Pine Barrens and a regular, upland forest, deciduous trees.
Yeah, and this was in particular found pretty much right next to the entrance, um, which definitely has a bit more of an upland feel to it because you're kind of slowly going down as you get to the boat dock where we always meet.
Right. Yeah. Marisol showed me the spot where she found this. This was not too far away from the main road that led into the park. The highway, the local highway. I forget the name of the road. Yeah, very surprising. I certainly did not expect to find that species there.
But yeah,
Cool.
cool mushroom. Either way.
Yeah.
Yeah, these can get quite big.
What is the species name? Sorry.
Rugosiceps.
Okay, thank you.
This guy here.
Yeah, Rugivo's cap, Rugivo's head. So that's an oak associate.
Nice.
All right, so let's see what INET has for New Jerseys. These are, the top two species are both European, right?
Yes.
European names.
Yeah.
So then we get vulpinum and longicurvipes. I wanted to look at some of these. Because one of these was Sigrid's and the other was Tom's. What do we think about this guy from far northern New Jersey?
Yeah, we have collections from Stokes. Actually, I have two, both sequenced. I think I got both ITS and TEF1 for them. I give up on them. I think they're vulpinum. They were growing in a hemlock grove. There might have been birch in there, or maybe pines. I forget. It was a mixed habitat. So this is an old collection. Any information on the habitat? Probably mixed. Yeah, this is probably like Wawayanda. Or maybe this is close to Green Turtle Pond. I think it is.
Yeah,
Passaic
Green
County.
Turtle Pond is right here.
Okay, yeah. So this is just to the east.
Yeah.
Yeah, this looks more deciduous area to me.
Yeah.
And she called this what, insigne? Yeah, I know. She might be right.
Right.
Not going to
We
argue
don't
with
know
that.
that she's wrong.
Chances are she's wrong, but she might be right.
And this is Tom's. This is up closer to Stokes.
Yeah, I see hemlock branches in there. Twigs, I should say. Needles.
Yeah, right
here. Chances are this is vulpinum, maybe.
Okay.
I don't know. I mean, there's also birch in there, probably. Birch leaves or beech.
These look like, I guess well, I don't know. Maybe birch. There's probably some birch in here if there's some luck.
Yeah,
right.
he might be right.
We have one photo of Versipelli, just a single photo.
Most likely misidentified.
Maybe like
I mean, I would not suggest a name other than Leccinum, section Leccinum.
Leccinoideae?
Leccinoideae, that's just too broad.
Too broad?
Yeah, Leccinum, section Leccinum.
We can do sections.
Yeah, that's the problem with INAT. We cannot do sections.
I think they have that option with section Leccinum.
So here we go.
There it is.
Leccinum, subsection Leccinum.
That's close enough. Yeah. It's a mixed habitat. Versipelli probably is birch associated. That would be my guess.
Here's the only arenicola observations from South Jersey.
Yeah. Remember, these are growing in the dunes, right like on the beach. And just imagine the ocean coastline and 30 yards, away from the waves, you have these huge piles of sand and some grass is growing in there. This would be your ideal habitat for arenicola. This is not arenicola, most likely. I think it's just a regular vulpinum.
It looks like the habitat is sand.
Yeah, there's no beach grass. Right.
Yeah.
Okay.
Yeah. This was found with pines, pretty sure of it.
Yeah.
I mean, do you have like an exact location? Which of those pins is?
It's somewhere in this purple pink box.
Yeah, well, yeah. I don't think this is,
unless it's down here.
I don't think so.
But this is the one that we might that name is the one that we potentially could have associating with those graphics, right?
Yes.
Yeah. We don't really have any forays in good coastal dune habitat. Not a lot of people looking.
You must be away from the trees and you must have grass and sand dunes.
Can we look at that snellii? Is that snellii? Is that a typical staining?
Yes.
So
that's what it does.
These thumbnails are for the species, not necessarily one of the New Jersey records, but
gotcha.
Yeah, so that particular photo.
Yeah, that's maybe a little more vivid than usual, but that's what
New
they
Hampshire.
do.
Yeah, I have a collection from Stokes that stains like that over time, so I can show it later on. But yeah, either snellii or variicolor.
Cool.
So we've got, well, this is the one that we looked at before. That was Lila's. We've got one other New Jersey snellii from way up by High Point.
It's closer to Wawayanda, maybe. Yeah, this is species New Jersey 02. That'll be my guess. That's the relative of snellii. And that's going to stain bluish. Oh, it stains red as well. Yeah. Oh. Is there a good photograph of the cap? See, it's lighter colored and has dark fibrils. So that's the hallmark of New Jersey 02 in my experience. It can stain red, but I only have one collection that does that, and that's confirmed by ITS. The ITS for those species is very stable.
Atlantic white cedar, spruce, hemlock, and some hardwood.
And the only tree that matters is birch. So it's a wet habitat, right?
Yeah, it looks pretty. Yeah, I see Bazania.
Okay.
So this is going to be in the cedar swamp.
So who knows? Maybe this is variicolor. Could be. Well, I don't know. Again, sequencing here would have helped a lot.
Right.
We now know what the ITS of snellii is because of the sequenced holotype collection.
Yeah.
But I would not exclude the possibility of snellii here. I think chances are pretty good.
Right. Yeah, let's get a mushroom observer. There's some that I wanted to look at over here. Let's see. No palins.
Zero. Oh, one. Okay.
Pittstown.
This is albellum.
Albellum? Right.
I think so.
Come back.
What do you think, Dave? Yeah, looks like albellum. Dave, you are muted.
Right. Thanks. Yeah, I would not disagree with that. It would be nice to see one sliced, but yeah, it's probably albellum.
Yeah, corrugated cap, cracking, oak.
Oak. Oh, okay.
Yeah, this is.
Does it say oak as for the habitat, or
Well, I mean,
I
I
see
think
the
I
oak
saw.
leaves.
I think I saw oak leaves in there. Yeah.
Yeah, I see the oak. I see oak
Yeah,
leaves.
gluten appellants. I mean, if you ask me what that species looks like, I would say I have no idea.
Yeah, I don't know what that is.
It's one of those cryptic names that Smith gave to one of his collections from Michigan, probably. Let me look this up, where that name comes from.
Glutenopalins.
How do you spell the second half of the word glutino?
Eallens.
Oh, two L's. Okay. Yeah, it's a Smith species, 1967, Michigan. Yeah. I don't think the Besets have it in their book. And if they do, it's probably their own interpretation of what that species might look like. Most likely.
Because it says that that's what he used. Let's see, 247. Yeah, they have glutenopalins. They get a photo of it.
Allegedly.
Yeah. All right, since glutenopalins, let's look at. Oops, it's not. This is one of Dave's.
Yeah, you wouldn't find a single observation under that name. That's mine.
Oh yeah, from Haynesville. Yeah, from northern
Right.
New Jersey. So I don't know.
Yeah.
Maybe it's just insigne. There's a lot of poplar there, but I ended up calling it pseudoinsigne. See, I think insigne is actually a, a Western thing, a Rocky Mountain thing.
I think it's a species from Michigan.
Oh, okay.
Let me take a look at Index Fangorum to see where the type came from. Um, it's a
So honestly,
Smith.
oh, go
Yeah,
ahead.
it is. It is Michigan. Yeah,
Well, a lot of things
associated
in
with.
Michigan are found in the Rocky Mountains. Also, there seems to be a lot of overlap. Even though the
Well, possibly it says here associated with Populus gradidentata. So this is your
Big tooth
usual
aspen.
aspen. Yeah, tooth aspen. Yeah, grandi means great, great tooth,
Yeah, big tooth.
big tooth. Yeah.
yeah, so I called this pseudo. I probably have a sample of this dried out.
So, it's an interesting one. Couldn't find that name applied to any other observations for New Jersey.
Yeah, it's just a name that not many people use, and I'm not sure I'm using it correctly.
Yeah.
Yeah, it's the common thread running through a lot of this tonight is these, especially
Right.
these section Leccinum species are really hard to pin down to species.
All right, and then we've got some
You contributing your opinion
subgranulosum.
about the mushroom?
Zilch.
Leccinum. That might have been applied at a 4A. It might not be something on MO.
Well, you should look for a name. You can search a name and see what observations are associated with that name. But I guarantee you that none are from New Jersey.
Yeah.
What's the name you're discussing now?
Sub
I'm sorry.
subgranulosum.
Oh, okay. I think I proposed that name once a long time ago, and Walt Sturgeon came on and said that I was wrong. So I, I don't think I've used the name since.
Subgranulosum is another species from Michigan by Smith and Thiers. Yeah, I'm not even sure what would that be, section scabra or section Leccinum. Uh, but it was probably sequenced. The type collection probably was sequenced by Colin Damnauer.
Okay, and then I thought we would look at some things on iNaturalist that are only identified to genus. And a lot of these are just single photos, but you see in the bottom left-hand corner it shows the number of of photos. So I usually go through and I try to find the ones that have the highest number of photos because it gives us the most to work with. So this is a Pine Barrens observation.
Yeah, this is our lovely vulpinum-like species.
Vulpinum group.
Yep.
Right.
Wow, look at that stain. That's
Yeah, I saw some
It's like
collections last
fuchsia,
year
sort of.
that stained really, really darkly blue. It was just amazing. So this is, again, a highly variable feature. But they would go for the pink stage, and then you get this fuscus color, which is grayish purple that you see in like those smaller fruiting bodies. Yeah, yeah, that color there in the cap. This is what Smith and Thiers called fuscus.
cool.
And yeah, the pink is going to fade away to this dark brown. Yeah, so it's all over the place. You cannot really map it onto any species.
Cool.
But
Yeah.
the best name is vulpinum, right?
Yeah, I would say yes. I mean, the thing is, chances are this is vulpinum, or if we have multiple species in that particular subsection that are associated with the pine, yeah, vulpinum group.
Right.
We'll throw our friend Kira a bone.
Yeah.
Oh, I see something in there. Never mind. That was species VA01. Yeah, that one on the left.
Here.
That's that species Leccinum or Leccinum VA01.
So what does luteum look like? I think we haven't actually seen one.
I'll
Yeah.
show you the luteum. I have a confirmed collection by DNA with a holotype.
Ah, great.
VA01.
Yep. Should have been New Jersey one or whatever. Where this is from, by the way? Do you have the state?
It's
Oh, it's New Jersey.
all these
Ringo's.
are New Jerseys.
Okay,
Yeah.
okay, these are all in Jersey. Ringo's. Yeah, I know where it is. Yeah.
Yeah.
It's near 31. Look at all these massive sectional exsinum. Unbelievable.
Yeah.
Yeah, they probably most of them came from the Pine Barrens.
Yeah.
You see these flaps on the cap? Yeah, especially the one on the right, the next one, next door. Yeah, irregular, very long. Yeah.
Mhm.
So that's a hallmark of sectional exsinum.
What about that dark gray one that went
Yeah,
by?
the one above.
That one looks interesting. It looks like it has the flap.
Yeah.
Maybe.
Yeah, this one. Let's take a
This
look
one
at this
right
one.
here. That one looks
This
interesting.
is Stokes.
Probably snellii.
Yeah, it probably doesn't have a flap. It just kind of looks like it because of the
Is
way
it a
the
single
cap is.
photograph?
This is the only one.
Yeah, what does he say? Just Leccinum?
Yeah.
Yeah. Yeah, I think chances are this is young snellii.
So here we can do CF snellii.
Yeah. That would be a great application. By the way, I have a paper that explains the difference between AFF and CF. It gives actually definitions of and situations where those prefixes, or how you would what we like to call them, descriptors, are properly applied. I have to dig out that paper, but I'll find it.
Yeah, a lot of these young. Look
Yeah.
for the pine needles in the background.
Yeah,
You
that's
look at
how
the
we'll find
mushroom.
them. Yeah. Pine barrens, or yeah, maybe stokes. What's the one on this? Yeah, this row, the one on the right, extreme
Yeah,
right.
that pink looking one.
Yeah,
one?
that would be probably snellii. Yeah, this one. Yeah, let's take a look
There's
at
another
this.
one though
That's
that
Emily's.
That's Emily's. It's huh.
Oh, I heard my name. Yeah.
Yeah, yeah. Wakey, wakey.
Yeah. Deep in emails.
So can you tell us about this collection, Emily? If you can still recall, this is 2021 October.
2021.
Parvin?
Parvin. Oh, which trail would I have been on? Boy, I'm not sure. There's so much at Parvin, and I go there frequently.
you can see the blue stain starting to set in on the
Yeah, this is section
Right
scabra.
here.
site.
Yeah. This is definitely section
Oh,
scabra.
yeah.
Oh yeah. Uh-huh. Bluing down
You
there.
see a bit of the pink too, pink
Mm-hmm.
red.
Yeah.
Yeah, we don't usually find snellii in this area. I think it's more of a birch hemlock species. So they find it a lot. We find this in northern Jersey. I haven't seen these things in South Jersey.
Yeah, very common where I live.
Yeah.
Very common.
You wouldn't happen to have a specimen, Emily?
No, I wouldn't. I mean, this was so long ago, and I wasn't really saving much at the time, so no.
Right. Got it. But if you recall where you found this, if you can go back and look for this again, that would be nice.
Yeah, I think I might know where it would have been. When did I find end of October? Okay.
Yeah, you found this on, end of October, which is typically where we find all these weird New Jersey 01, New Jersey 02,
Mm-hmm.
Indiana 01 species, and FREC, which is Forest Resource Education Center, and holopus also there. Yeah. So yeah, they do come out, in the fall with birch.
Emily, when you go to Parvin, do you normally go around the lake?
So
Or I guess that's a lake. Is that a lake?
yeah, it's a lake. So there are two trails that I typically go toward. Yeah, one is the green trail that goes around the lake, and that's pretty long, and there's a lot of different types of
Mhm.
environments through there. And the other one, there's a parking lot on the other side. Then it's
Yeah.
basically in the woods, and that one has some interesting stuff. I think that's the Orange Trail.
Oh, cool. I like Parvin. I've never gone back on the parking lot side, so that's good information.
Yeah, there's a lot of trails there. Some of them aren't open, but if you get a map, it's more than you think is there. There's a lot.
Would this be a good park to go to with the club, or it's too small for a large foray?
There's enough parking for sure.
Yeah, there's enough parking, and I
And
would say
but you have to do a loop. Like if you go around the lake, you have to do either a full loop or double back on yourself.
that's a pretty
Which
long loop, though,
exactly.
especially
Uh,
if you're mushroom hunting. Like I've spent 5 hours on that loop before.
yeah, yeah. But I don't know about the other side, so maybe that's a little bit better.
So what I would say is Jason? Yeah, Jason's still here. You're in the area. You've probably been to Parvin a bunch. What's your thoughts?
Yeah, I think the nice thing about Parvin, it's got a beach, I mean,
Yeah.
this whole complex here, I mean, it's got, a nature center, and it's got big bathrooms, and, I haven't been here in the summer, but I'm guessing this beach gets a lot of summer use. So if we wanted
Mm-hmm.
a foray that has a lot of, for targeting mostly outreach, or, setting up tables in an area where there's a lot of traffic, that might be a good spot.
That's true. And there's a lot of other areas that have tables as well throughout the park.
Mm-hmm.
So Jason, would this be the inner coastal plain or the outer coastal plain park? or area?
Outer. I would call this probably outer.
Okay, and this is Salem County, or what county is this?
I think it's Salem, maybe
I think it's
Cumberland.
Salem. It's right on the
I think
cusp
it's
between.
I'm pretty sure it's Salem.
Yeah.
Yeah, we don't have forays in Salem County, never had. I think it's kind of a tucked away area, and very few club members are around, so it would not be logical to go there with the club because, for a lot of people, it's going to be a very long drive. Even for me, it's a pretty
Yeah.
long drive, maybe like 55 miles, I would say.
Yeah, Scotland Run is going to be up near Glassboro, so it's
Right.
pretty much this far further south than.
Yeah, I know it's 62 miles to Belle Plaine, so I would say maybe 50 here, or at least 45. I know it's an interesting place. I mean, I would like to go there sometime and take a look.
Yeah, I'll just make a note. I mean, Jason, you and I were there one time for a different club, Bio Blitz, and we found a bunch of mushrooms. I mean, it was pretty good conditions, but there were a lot of mushrooms there.
Yeah.
Yeah, if it gets a good rainfall, you'll find a lot for sure.
Yeah, it's an easy drive. You just take 295 and then 42 and 55, and you're there. I mean, I basically take that road when I go to Belle Plaine. And this is your backyard, Emily, right? Pretty much.
Yep, it's 15 minutes away from me.
Yeah.
Yeah,
Well, you're the president now, so you get to throw your weight around and have some forays that are close to where
get
you
one
live.
in your backyard. Yeah.
That's
And
good to
Igor,
me.
there's an even better drive than getting on the highway. There's a straight shot if you get on 561 and take it down that way. I don't want to get into that, but
Yeah, 561, I'm not sure. It's probably one of those smaller local highways, but I would see like 55 and then maybe cutting. There's an exit. What's the name of this road? I'm trying to see. It's called Buck Road. And there's an exit off 55 onto Buck Road, and you take it just straight down and it just goes to that park.
Yeah, that'll work. I know where that is. You can actually if you're going to Scotland Run, you can get off at that same exit and just go a different direction. It works for either one.
Right. Yeah, it's next to Pittsgrove, so yeah, it's an easy drive. It's just probably going to take probably 45 minutes to get there. Cool. Did not even know about that park. So what else do we have? So Dave was talking about another collection from here that he was interested in. Which one was that, Dave? You're muted, by the way.
Oh, yeah, I just unmuted. It was on the far right. It might be up a little higher. Yeah, go up a little higher.
Whoa. Can we take a look at this one that says Leccinum with 3 pictures? I don't think this is Leccinum. Yeah, this one, yeah. Before we go to Dave, just quickly.
It's going to be oliveisporus.
Yeah. Where was this found again?
Not far from north of Vineland, kind of between Parvin and Scotland Run.
Yeah.
Is this Porphyrelis?
No, this is Rufa Boletus, I think, probably. Yeah, this is oliveisporus, I would think.
Oh, okay. Yeah, I don't get that around where I live.
Yeah, it's a outer coastal plain thing. So
right.
Yeah. I know it still calls it a Boletus, but yeah.
Well, Rufa Boletus is it's going to be called that when it gets published under a after a generic transfer. But yeah, for the time being, that name is still applicable. Is correct, but obviously it's not a Boletus in the strict sense.
Yeah. Oh, that still wasn't the one that Dave was.
No,
I'm
babes
I'm
is
not
above, right?
sure. It went by really weak, really quick. Oh, that one on the right. See in the
This
right
one?
hand, right? Yeah, this one. The one that's got like the textured cat. No, the below that.
Below that. Oh yeah, this one. Yeah,
Probably albellum. This is albellum.
Yeah, I guess so.
Yeah.
I'd like to have a look at that one though. It looks kind of. Oh, it probably is albellum. Yeah,
Corrugated cap. The
yeah,
skin is
yeah.
very
Okay.
glabrous and ivory-like, so
Yeah,
has
I
that
agree.
sort of
It's
yeah,
probably
yeah,
just albellum. Yeah.
yeah.
All right.
cool.
Corrugated, and then as it expands, it just gets cracked and
Yeah.
gets rhymose.
Yeah, that's typical albellum stuff going on there. Yeah. Okay. And this one, it's just a spread out red or orange one. It looks like the flap is. It probably should have a flap, but it looks like the cap got really wide and it fell off.
Oh, it's still there.
This
Um,
one or this one?
that really big one that was just on. Maybe it's that one. It went by kind of fast.
Or oh, this one.
Yeah, let's look at that one. That looks interesting. It's probably
It's just your typical section Leccinum, and there you go.
yeah. So it's just like vulpinum group probably.
Which area is this? Chatsworth?
That was near
Okay.
Bat. It
Yeah.
was near Badstow, I think.
Best. Okay. Yeah. That's your Pine Barrens.
Yeah. Yeah. Let's see if there's anything else that.
Yeah. If you scroll down, there
Jumps
was
out.
one interesting collection that had a kind of a yellowish upper stipe. Can you just keep scrolling down slowly? It was, I think, the third one or the second one on the right, from the right. Below that, oh no, it was on
No,
this
let's see,
page.
yeah.
Let's go up.
Here?
Yeah, this one. Yeah.
This is Delaware Water Gap.
Yep.
It's like up by Tammany.
Northern New Jersey. So I think this one is New Jersey O2, which is a relative of snellii. Yeah. Didn't cut them. Yeah. So as you can
Right,
see, putting your Leccinum to a section is very easy. Putting assigning species names is going to be exceedingly difficult without prior DNA work.
All right, what do we think about this guy?
Section scabra. Can we see the cap?
Yeah.
Yeah, just could be that New Jersey O2 thing. Yeah. So where was this found? West Milford? Okay.
Way under.
Wawayanda, yeah. Yeah, there's
This
birch
is at
there.
the way under foray in 2020. This is one of mine.
This is ours or New York's club foray?
This is my this is one of our forays.
Oh, our forays. Okay, got it. Yeah.
Yeah.
Yeah, you see it's just a variation of the same theme with these section Leccinum.
Yeah.
So what's the one on the last row, the one on the left? Yeah.
This one?
Yeah, just curious about this one.
It's like one of Luke's.
By the way, Luke's Boletus in his avatar, the thumbnail. This is Boletus biliae.
Yeah, look at that.
It's a rare species described by Ernst Both from Cape Cod. Yeah, just another vulpinum-like entity.
Mm.
Beautiful species.
And I love this mushroom as an edible, personally.
Yeah.
Yeah,
I have jars of that stuff.
I like to slice them fairly thinly and fry them in butter until they start to get brown and a little bit crispy, and you salt them, and they're like sort of like bacon.
Yeah. In my experience, brown is the wrong word. They turn black
Yeah.
before they.
But then they go
caramelize.
back though. Then they go back. They lighten up after that if you keep
Yeah.
frying them.
Yeah, that's true.
Yeah, really good.
One thing I want to.
I gave these to a friend one time, and he was like, "Dude, when I cut them, they turn black. I'm not eating these."
Yeah.
And I had to assure him they're good to go.
So good.
Yeah, if your
Compare.
arenicola is growing with trees, then it's not arenicola.
Some of these, I'm looking for not necessarily New Jersey, but just some type of high quality observation. You can see a lot of these are growing more with Hudsonia than anything else, which is another dune species.
So what is this thing that you called Hudsonia?
Hudson, yeah, the Pine Barrens heather, this little short shrub thing.
Yeah, well,
That
heathers
grows
are,
down.
arenicola grows with these things too.
Yeah, I mean these are.
So, but this is from like Newfoundland or something, right?
Yeah, but
Yeah,
yeah,
Prince
definitely
Edward
coastal.
Island. Okay, that's believable.
Yeah.
I
So
mean, I
the
don't
shrub
see dunes in there, but yeah.
the shrub that you're talking about, Jason, that's a small, like very low to the ground growing
Yep.
thing.
Yeah.
And that's also
It
amongst Cladonia, right?
right.
If
Cool.
you
But
look at
a
the
lot of
notes, yeah, sorry to interrupt. If you look at the notes, they do say growing with Hudsonia on stable gray dunes.
mhm.
So chances are they probably have it correctly identified. Can we go into the other pictures that show the cross section and the discolorations?
And they got a DNA blast
Oh,
as well.
yeah. What do you think you can copy and paste the link to this observation into the chat so I can
Sure.
take a look at this later on and see if it fits the ITS of arenicola?
Yeah.
If you can still separate them from the rest, because this is going to be section Leccinum. Thank you. Bluing on the stipe, yeah, which is very typical of
Mhm.
everything.
And a little bit around the cap margin as well. This is this mycorrhizal position with pine trees. Does that sound right?
That's incorrect.
Yeah. This is, it's straight out of the Hudsonia. It's all Hudsonia around it. This is probably the same location, same observer. Oh no, this is different spot. Still pretty coastal though.
Do we find Hudsonia in New Jersey?
Oh yeah, well, over our coastal dunes, Island Beach State Park, Sandy Hook. We've got two species of Hudsonia. One is more common in the pine barrens. They both occur on the coast. One's goes further inland than the other. We got the beach heather, and we got pine barrens heather. This looks more like the beach heather, tomentosa, and they hybridize. Actually, this kind of looks like two side by side. One's got kind of whitish tomentose hairs on it, so it looks paler than the one ericoides that's more common in the pine barrens. This is the plant that what's that? There's a genus. There's a rare mushroom at Caddis Island that some expert came to New Jersey and found a specialist with Hudsonia. But I can't think of the mushroom genus.
When was this?
2017-ish. John was telling me about it.
Oh, John Burkhardt?
Yeah, he's asking me about Hudsonia. It's on the tip of my tongue. I'll have to look it up.
All right, so I grabbed that sequence from this alleged arenicola. And by the way, that is probably mostly ITS2 because I can see the beginning of LSU and I don't see it's a fairly short sequence. So I think most of the, if not all the ITS1 and 5.8S region, which is between ITS1 and ITS2, are missing. So this is ITS2 sequence. And when I blast it, I get 99.73% identity with Leccinum piscinum from Austria. And at the same time, it's 99.73% similar to Leccinum species Indiana 02, Steve Russell. So there you go. Not very useful.
Indiana 02, yeah.
Well, does that, I mean, that seems very similar. The from Austria and Indiana. So does that prove we have the same species, at least, whatever you want to call it, from
No, it
Austria?
just proves that all these Leccinum, their ITS sequences are pretty much very similar, especially the ITS2 region, which is much more conserved than ITS1. So you will not be able to distinguish between individual species when looking at ITS2, probably, most likely, in that section, that is. So it's kind of a generic, very generic barcode. It's just not specific to any species in particular.
Got it. Thank you.
Yeah, so you have Austria, you have Newfoundland or Prince of Edward Island, and then you have Indiana. And what's the common denominator here? Well, there's no. They're just from the northern hemisphere. Different continents, different regions, different latitudes, different ecological zones. I don't know what else you can say about them. So, yeah.
That genus that was on the tip of my tongue is Hebeloma. I think there's some Hebeloma species that associates with Hudsonia.
Well,
that information would be at Henry Becker's website, which is hebeloma.org.
Right.
You just have to search Hebeloma and Hudsonia, and maybe with some luck, you'll find it there.
Indiana has a lot of dune areas.
So this is Cape Cod, right?
Yeah. Coastal dune habitat.
Oh, look at that.
Yeah.
That's beautiful.
Like I said, I'm not denying the existence of arenicola. I'm just saying that with ITS, you would not be able to make any assignments, especially ITS2.
Right. All right. Anybody have any last minute shares or questions?
I can show you a few of my observations if anybody wants to see them, especially the ones that have been sequenced and have a confident identification attached to them.
Take it away.
Well, let me share them. Let's see. Chrome tab. All right. So here we go. This is all arranged by date. So I'm just going to... This one here is your rubropunctum. And this is Shark River County Park. So we've seen that before. Albellum. Yeah, this one I haven't sequenced it yet. Also found at Shark River in July. Not sure what it is. Oh boy, this is an interesting species, and I have an ITS, except that I haven't processed it yet. It has some difficulties. But look at this presentation here. You cannot even see... it doesn't do that. You cannot even see scabers. You see like reticulations or a pattern resembling reticulation in this species. And again, I think there's at least two collections here. By the way, this fruiting body here on the right with a dog-legged stipe, that was a mistake. That's probably albellum, dark one. So this is a different species. But these four here, there are at least two separate collections, but they're all the same species. This is their caps, and this is how they stain.
Oh, wow.
So you see it's yellow here, and usually in the Leccinum section scabra, when you have a stipe with yellow stains, that area is going to stain blue, almost certainly. And that's the final color. So all the red and blue, sort of whatever bluish tinges you had in there, went away, and it just became this grayish brown, or whatever you want to call it, violaceous brown. So this is all the other fruiting bodies cross-section at the same time and showing the same pattern of staining. Sorry about the rug. Yeah, I was doing this like at 10 o'clock at night or something. So yeah, might be species Indiana 01, but I have to work up the ITS more carefully, the chromatograms, because this was Sanger. This is that aenigmaticum, Hemileccinum. This is a collection from Smithville that was sequenced, but I already knew that before sequencing. So has all the hallmarks of Hemileccinum. But it's interesting that the ITS would not place this into Hemileccinum. The ITS sequence is very unique and unlike anything else in Boletus. So I had to sequence step 1 to get the genus assignment. Beautiful species. No staining, like in all Hemileccinum. Corrugated cap, sort of a kind of a reddish brown. Not quite sure how to describe that color. Kind of a rich, yeah, I'm just bad with colors. Anyway, so that's that. And there's going to be more collections of it. So this one is luteum. And that's a gospel. That's gospel. That's what luteum looks like. And that's the staining. Note how beige or yellow brown the flesh is below the cuticle. This is not staining. This is how it was when it got cut. And that's how they stain after a while. sort of blackish brown over time. Can you hear me? Anybody?
I can hear
Yep,
you.
we can hear you.
Okay, yeah, it was
Yeah.
just silence. I thought maybe I'm just talking to a void. So this is luteum. Yeah, and this is that rugosiceps from Scotland Run, a very robust species. I don't have a, yeah, I do have a scale. So this is me holding it in my hand, so it's pretty large. I think this is Maricel's collection, and I did sequence it, and here's a discussion. I'm not going to go through that. There are some matches from Indiana and North Carolina at about 99%. So it's probably the same thing.
This is one Brandon showed too. Did he show this too?
I'm sorry, what?
Did Brandon show this as well?
Yes, it's
Yeah.
from the same location.
Oh, okay.
It was on the same, yeah, so this is September 2022, and Brandon just found maybe a different cluster. But this is a collection by Maricel, and you see it does stain eventually. Sort of a, not quite sure how to describe this color. What do I say about that here? I do say it was a surprise because it's in the Pine Barrens. Yeah. Probably reddish. I think it's stained reddish, fading away to this dingy brown color. Yeah. So that's probably rugosiceps, possibly. This is albellum. Different collections. Okay, this is not one specimen. There are multiple collections here. Several specimens found by various foray participants. So you can see how variable it is. It's not quite white, but that's what these things look like. They can be quite dark. This is also from Scotland Run. This collection here is the sequence collection of what I believe is subglabripes. This is from Stokes. Growing in mixed woods, oak, birch, hemlock, and pine, and maple near the Stony Lake area. And you can see, it kind of resembles rubropunctum, except that the color of the cap is not as red-brown. It's a different shade of, it's not red. It's some shade of brown, kind of light chestnut. I'm not sure how to describe it. But when you have those two species in your hand, you will be able to tell the difference. They are not the same, but they are indeed quite similar looking to each other. So that one was sequenced, and I'm pretty sure that's what this is, because there are sequences from Maine and Vermont that match that, and I think that's the one that has to be subglabripes because it was described by Peck from upstate New York. Yeah, these are various. Yeah, Steve called it Indiana 02. That's from Stokes. And next to it is from FPP. So yeah, we don't have to talk about these. It's just pointless. So this is what holopus looks like. I don't have it sequenced, but I'm pretty sure that's what this is. It's very pale and was growing near water in wet conditions. And it stains red. See, it doesn't stain immediately when you cut it. what does it say, 30 seconds after cutting, and this is after 20 minutes. So holopus likes to be in a very wet environment, like boggy area. This is another aenigmaticum. This is from Washington Crossing State Park. I have this collection well sequenced. TEF1 and ITS, I think. This is more holopus also from the same location, just different year. Forest Resource Education Center in Jackson Township. We didn't talk about hortonii, which is a very distinct species. It has a very corrugated cap, but otherwise looks like your typical Hemileccinum. We had several collections from Stevens State Park from a while back before we stopped going in there. This one actually is holopus from the Pine Barrens, even though it's dark. This one got sequenced. And this is albellum from actually my property. You see how dark it is? The caps are sort of this reddish brown color, but it was growing under beech and oak. So what else can this be? Corrugated cap, no staining.
So Igor, have you seen this species or this specimen come up multiple years, and is it consistently dark?
No, I only seen this once. This is my property. And that area has a lot of moss and very hard ground underneath. So it has to rain a lot before all that moisture sort of seeps in and all these mushrooms pop up. But from the same area, near the same trees, which is, red oak and beech, I found Tilapilus, Boletus in there. I found Lamaua pseudoinsigne, and I found Porphyrilus sordidus, all growing with these trees, and also this thing. I only saw it once, and I was back in 2018. And I look at the date. It was that exceptional year. It was very rainy and very warm. I mean, this is unusual for this species to be fruiting so late in the season. I mean, this is like mid-October.
Mm-hmm.
And this one we didn't talk about. This is Visidicarugus, longicurvipes. I just have one photograph because this is such an easily identifiable species. Orange yellow cap, very sticky, stains bright red with KOH, has a long and S-shaped stipe. There's nothing else that looks like it. So once you see it, you'll never forget it. And that's a nice collection of rubropunctum. So you can appreciate the darker cap color, but if you recall, subglabripes looks very similar, except that it's much lighter in the color of the cap and has much less red in the stipe. These puncta are very, very red. And these are the caps. These colors are very accurate. Dark reddish brown. And that's what the surface looks like. So you see these scruffy points, cuticles, whatever you call them, and here it sort of gets kind of textured, but it's mostly red. And that's a typical look of the ashtray bolete. So you can just tell the two apart by the intensity of redness in the stipe and the lighter color of the cap for subglabripes as opposed to rubropunctus. And they both have yellow pores, yellow tubes, and they don't stain blue or anything like that, or any other color. This is the same as we saw before from Scarlet Run. I believe this is the same species. This is Leccinum or Leccinum rugosiceps. This was found in Somerset County, a location that I keep secret from other people. It comes out every year growing in short grass and moss under big oaks. It's a private property I have permission to collect on.
I missed the species. Which is it?
Rugosiceps.
Oh, it is another one. Okay, thank
Yeah.
you.
I think I had them sequenced. Not this particular collection, but a collection from another year at the same location. And I may have an ITS sequence for that. I haven't looked at it in a very long time. Yeah, so this species is Indiana 01. So that's a rare one. I cannot really say much about it, except that it stains both red and blue in the flesh. And in that way, it resembles snellii, but the ITS sequence is very different. And hence the designation Indiana 01, because Steve Russell happened to find a 2, and he gave it that temporary code before I even came up with the idea of having temporary codes for fungi. But it's certainly a rare find, not only in New Jersey, but in the United States. Not many collections. And this one is New Jersey 02. This is a relative of snellii. Multiple collections from Stokes. I all believe they're the same species. You can see the caps are fairly light brown, and they're covered with these fibrils, which are much darker, giving it like a mottled appearance. I'm probably going to have a better picture of the caps. You can see the caps here. You see the fibrils, but the background color is very light. And they all stain blue in the bottom portion of this type, but there's no red. Yeah. Very common species. Probably across all eastern USA. And this species here also came from Forest Resource Education Center, just like holopus that I showed you, just like Indiana 01 I showed you. So apparently there are multiple species growing under the same trees in this wet area. There's like a small pond some distance away from the nature center. But that's New Jersey 01. It has a different ITS from everything else. And it's a young specimen. Dark scabers, and the flesh is going to stain both, I think, blue and red, mostly red. So apparently we have several taxa that can have both blue and red staining. It's very common in section scabra. And this is my alignment of ITS sequences. I was just comparing different collections, different sequences from GenBank. And GG80 Kata is the very end of SSU. So everything in blue is ITS. And you can see that the species has consistently, very invariable ITS sequence. So ITS barcode for section scabra tends to be very good. There's one ambiguity here. There might be gaps because of you have several T's in a row, which is very common in anywhere. And you have some jitter here. But by and large, you can see that there are multiple sequences that are essentially identical to my own collection. This is snellii. This was sequenced. And you can see the difference between New Jersey 02 and snellii, that the cap in snellii is much darker and has areas that are white, almost white, so it kind of has a mottled appearance. And the staining in the species is distinctly blue and red. This is over time. So this is 15 seconds after slicing. This is 32 seconds. And this is just over 2 minutes. And this is 5.5 minutes. And some old tree that I built in GenBank. So again, I measured the spores, I got the sequence, and I did confirm it to be snellii from the holotype collection eventually. And this is subglabripes from Vermont. At the nymph, we collected this. So that's what that species looks like, growing under birch. So no red in that stipe. It's essentially all yellow, unlike rubropunctum.
And it's not as scabrous. Is that where the epithet comes from? Subglabripes?
I think subglabripes refers maybe to the cap texture.
Oh,
I'm
okay.
not quite certain. I have to look this up. But the stipe itself, I can probably zoom in. This is not any better, but you can see. I wouldn't call them exactly scabers. They're maybe like squamules or whatever. But yeah, they're a little bit scruffy.
Okay.
Yeah, but there's no red in the stipe.
Right.
Like I said, it's a very rare species in New Jersey. I think this is probably where it ends. If you go south of that, you would never find subglabripes. Even like in Delaware or Maryland, is just too far south for it. Unless you are at an elevation somewhere, like in the Appalachia, where it's colder and where you have birch, you might have subglabripes in there. This species here is also from a private property in Somerset County. I don't know what this Leccinum is, but it's not the same species that I showed you before, which was rugosiceps. This has a much darker cap color, and I didn't have the heart to pick them up. They were too young, and I just left them where they were. So unfortunately, I haven't seen them since then. But they were growing not too far away from that collection of rugosiceps. So another mystery. This is albellum. This is how white the caps can get. Well, not quite white, but are much whiter than the ones that you've seen before. And at the same time, you have a darker specimen just growing next to it. Highly variable species. So this is variicolor, all right? And this is something that somebody brought to the Como foray in 2016. It was found probably in Connecticut. Yeah, Union, by Robert, my friend from Connecticut. And you can see that the cap resembles that of snellii, dark brown with this much lighter areas. And it's stained both, well, it's stained just blue mostly, I think. But we got the sequence, and it matched a sequence from Europe. So this is, I'm pretty certain that's what this is. This is variicolor. And you can see a lot of people voted, I would call it that, just because of that ITS. Yeah, and look at that albellum. I keep going back to the species just to demonstrate how variable albellum can be. I'm not sure what color you would call that. It is some shade of brown, but it's almost like it's infused with pink. And that's the color that you often see in darker varieties of the species. Once again, you only find this under oak, and it doesn't stain at all. This is something that we found at a place that we only went to once or twice. It's called Palisades Interstate Park. In which county would that be?
Bergen.
Bergen. Yeah, this is close to the Hudson River. So you just have White Plains to the east, and this collection sort of looks like a hybrid between subglabripes and rubropunctum. And I called it subglabripes just because I thought there was just not enough red coloration in the stipe, actually almost none. It might be rubropunctum, and my idea is wrong, but at that time I didn't know that species smelled like, cigarette ashtray, whatever. So I decided to call it subglabripes, but I might be wrong about the identification. Unfortunately, I didn't save it, so there's no sequence. Yeah, so I think we're just reaching the bottom. Yes, so this is the first collection of Leccinum Virginia 01, and this was by Luke at Horseshoe Bend. And he said he said ash, hornbeam, and oaks. So it has a yellow cap and it stains black immediately when you touch it. And when you slice it, it stains reddish brown like that. And I think eventually that color changes to black. I don't think I recorded that transition, but certainly a very distinct and unusual species, whatever it turns out to be. Maybe something new that Smith and Thieres never described, and from Michigan because it just doesn't grow there. But obviously, it's distributed as far south as Virginia. We find this in New Jersey, and we probably find this in Indiana as well. I would not be surprised. So that's pretty much it. I probably have other things here, but this is just more albellum. I just keep coming back to the species because, again, it's so variable. You can see the pitted cap, very pitted, and a lot of brown coloration in the cap in this case. And the ornamentation of the stipe is a mixture of scabers on the upper portion, but it's also highly striate and kind of pseudo-reticulate. These ridges that are in the stipe, are forming this kind of a crisscross reticulated pattern. So you will see that in in albellum. Don't let it confuse you. So that's pretty much it, unless you have any more questions about anything that I have here.
I got a few more since you mentioned subglabripes is not super common. There's 16 INAT records, and a few of them have reddish in the stem. So I think those should be easy corrections.
Yeah, let me stop sharing, and you can go to INAT and take a look at them.
Yeah, let's take a look at some of these real quick. One of them was mine. I think I just corrected it, but yeah, this guy here.
Yeah, let's flip through the pictures. And you found this where?
Hunterdon County.
Mm-hmm.
But yeah, look at all that red.
Too much red, yeah. Probably rubropunctum.
All right, so I corrected that one. Here's one. Fortunately, smell is going to be an important thing for a lot of these. Don't have any mention, but it looks like a little bit of reddish brown here.
Yeah.
So
And
that
this
is
was
enough to
what's the area? I'm just trying to get a
Pemberton, Whitesbog. It's a big radius, but northern
Pine
part of the
Barrens.
Pine Barrens.
Yeah. Yeah. Rubropunctum would be, I'm pretty certain that's what this is.
I think these are all the same person, same day.
I'm not even sure this is actually Hemileccinum. It's a different bolete, I think, altogether. I think it's an Mawa. Probably Citizen Sibilis. It just doesn't look like
Mercer County.
Yeah, it doesn't look like a Hemileccinum to me at all.
You want to call it Boletus or Lamoa is the first species suggestion. So let's call these all.
Yeah. I mean, the picture's not that great, but from what I can see, like this one here, it just doesn't really look like, even if, say, rubropunctum were blurry, it wouldn't look like that.
Yeah.
The cap is not corrugated, it has a different texture, has a different color. It's, whatever this is, even if it's not Citizen Sibilis van Maua, it's certainly not Hemileccinum, subglabripes, or rubropunctum.
What about this guy?
Yeah, this is aenigmaticum. Judging by the color of the cap, I think. Let's take another look. Let me see more pictures if you can zoom in. Okay. Yeah, I would call this aenigmaticum.
I'm gonna call this.
Yeah, and that name is not an INAT, obviously. It's a it's a temporary name. It's a provisional name I created on MO, and you can basically refer that person to MO observations.
This one takes care of that row. Oh, this is the same Moa. Here's this.
Yep.
This is that Landmoa pseudosensibilis, the same one. For some reason,
Yes.
the photos are different.
Yes, it's the same. It's the same mushroom as the ones in the other observations. Yeah.
Yeah. Here's one from Nicole.
Nicole Engel.
Yeah.
So she's the swamp witch.
Yeah.
Rubropunctum. Too much red.
Yeah.
Way too much red for some varieties. And she found this in Thompson Park. Okay.
Nope, just one.
Yeah, this looks more like subglabripes. But but this is where this was found.
Is that
Belmar?
Shark River? It kind of
Shark River.
the
Yeah.
wall.
Is this your collection? No, not yours. No.
No. 2021.
Can we zoom in, please?
Yeah.
I want to see this type. Oh, let's see the cap. The types are just, one photograph is. I don't know. The caps don't look corrugated or wrinkled enough for this to be called. This may not even be Hemileccinum.
Yeah.
Not enough
we'll leave that
information,
one alone.
but I suspect it's not. Yeah, that's rubropunctum. Dark cap. And the location is
Great Swamp.
Great Swamp. Yeah.
There's a Tom Bigelow. Up towards Green Turtle Pond.
Yeah. Chances are this is a subglabripes, at least, geographically it fits. And it resembles my collection from Stokes that was sequenced. So that ID probably is correct. Oh, yeah, that's
a nice picture.
Yeah, that's definitely subglabripes. No question about this.
Cool.
Did you see those stipes? They're pale yellow, no red in them.
Yeah.
And that was found where also, Stokes?
Stokes.
Yeah.
All right, let's see how we're doing. Yeah, 10-7. Some of these are just.
Yeah, I would vote that subglabripes, 100%.
And Chester.
Yeah, Shiv Nature Preserve. Can we zoom in?
Is that
reticulation?
It looks like, yeah, it almost looks
It
reticulate.
looks like separans. Yeah, I think this is Boletus. That's a porcini.
So here's one of Shihong's. Also from
Helmeta, yeah. Yeah, rubropunctum.
Yeah.
Not much red though.
Yeah.
Yeah, this one is more ambiguous.
Yeah.
I mean, rubropunctum does not have to have like a distinct red stripe. It varies too.
Right.
Yeah, you know what? I would just leave it.
Right. Those from
Yeah,
this year.
this one is AC doocy.
Yeah. Yeah.
Can we go to this one? Yeah, because I think this is something else. No, not that one. This is
too far gone.
Too far gone. It's either this one here.
That's too viscous, right?
Yeah, this is wrong ID.
Yeah.
This is longicurvipes, I think. Look at that shiny sticky cap.
Yeah.
So I would call it Leccinum longicurvipes.
Very cool. Right, so we
yeah, this one is could be one or the other, and I mean subglabripes or a rubropunctum, but it's kind of old and too far gone.
Yeah. This one we just did.
Yeah, this one I'm pretty certain
16 down to 6.
Yeah, once again to emphasize, subglabripes is rare in New Jersey, and you're most likely to find it in northern counties like Sussex, Passaic, Warren, growing with birch in mixed woods. Once you go south, chances of finding it are pretty much nil. I recall finding this at Meadowwoods and Stokes, and that's about it. And I think in both cases it was growing under mixed in mixed woods with birch.
All right. Well, that's all I have time for. Let me stop the recording.